Good morning. It's Michael Mallinson of the Canadian Spondylitis Association, broadcasting on the Arthritis Broadcast Network via Facebook Live. This morning my guest is Dr. Nigil Haroon of the University Health Network, Toronto Western Hospital. So I'm just going to ask you to introduce yourself, Nigil, and tell us about your role in rheumatology.
Thank you, Michael. Thank you for this opportunity. So I'm Nigil Haroon. I'm a scientist at the Criminal Research Institute. I'm a rheumatologist and clinician scientist with the University Health Network, and I work as a faculty member with the University of Toronto as well.
Thank you. You've just given a fascinating talk on the immunology and pathogenesis of ankylosing spondylitis. Not being a technical person with no medical training, I'm afraid I didn't understand a lot of it. Conceptually I did. I wonder if in lay terms you can just tell me something about the immunology and pathogenesis of ankylosing spondylitis and what you're discovering about folding in cells and things like that.
Most certainly. Thank you for picking up that folding part. So it's quite well known that HLA-B27, which is a gene present in 85 to 90% of the ankylosing spondylitis patients, it's important for the pathogenesis of AS. But exactly how it leads to the inflammation that we see, how does it lead to, especially in the spine, why the sacroiliac joints, why does it end up in fusion in a certain proportion of patients? These are all questions that are wide open, we don't understand. So the B27 association was identified way back in 1973. That's a good 44 years now.
Yes.
Many centers have tried to look at exactly what is causing the disease, how does B27 lead to inflammation. So the best possible explanation that we have, we can narrow it down into two or three possibilities. One is that B27, it's a normal gene present in around 7% of Canadians, right? And the normal function of that gene is to present antigens. So, for example, if we have an infection, let's take, for example, flu infection. If the virus gets into the body, it can go anywhere in the body. And it's very difficult for our army of immune cells to find the virus because not all cells respond to that virus. Only a certain proportion of cells have specificity for that virus. So you can't go throughout the body in the bloodstream looking for the virus. Instead, what the body intelligently does is it has a separate group of army cells that grab on the virus and process it and presents the virus to the surface so the T cells can immediately identify it. So we have two groups of cells. One is that grabs and presents the virus, the other group of cells that identifies it and kills it. Okay? So B27 is the molecule that grabs and presents the virus. And perhaps B27 has, compared to other genes like B1, B2, B3, B27 may be identifying a particular virus or a bacterial molecule or some kind of antigen that is leading to the body to respond in the way that we see in AS. So maybe that immune response is a sort of an abnormal response to the B27 infection combination. May not be infection, we don't know that. It could be anything, it could be any kind of antigen. So that was essentially what I was trying to explain with that first B27 antigen presentation. The other explanations are interesting. B27 is not a very stable molecule, so it can misfold. That's what you were referring to, the misfolding. When it is not properly folded, it cannot present the antigen. So what it does is it just keeps accumulating inside the cell. The body tries to present the antigen, so it produces more of the B27. And it keeps accumulating, and finally the cell undergoes a lot of stress, produces cytokines. You're familiar with the TNF, TNF, interleukin 23. These are all the cytokines that the cell produces when it's under a lot of stress. So maybe these are the ways in which it leads to inflammation.
And you were also talking about bone formation and inflammation and the paradox of whether it forms bone or whether it destroys bone.
I don't think we fully understand it now. It's definitely a paradox. We have excess bone formation on the outside the vertebral bodies that leads to the fusion between the vertebral bodies. But if you look actually inside with the bone density scan, the bones are brittle. There is osteoporosis inside. So for a number of years it was thought that maybe you have a lot of bone formation outside, so that leads to sort of a decrease in the weight transmitted through the actual vertebral body. So the offloading of the vertebral bodies leads to decreased bone formation, because the more you load the bodies, the bone, the more the bone responds by increased formation. So maybe you're offloading the bone vertebral bodies, that's why there is inflammation, that's why probably there is osteoporosis inside. But in a probably better explanation is that there is local inflammation that is driving two processes. Within the vertebral body, it's driving bone loss because of the local environment that the inflammation is producing there. And on the outside, it's probably secondary to chronic inflammation that turns the bone into fat and finally into bone formation.
Oh, I see, yeah. It's fascinating stuff, and it's the work you do is amazing. Maybe you could tell me a little bit about one or two of the research projects that you're currently engaged in.
Yeah. So I'll talk about a paper that was published already, but we're doing more work on understanding that a bit more. So there is a family out of Newfoundland. It was they attend the Newfoundland Memorial University Hospital, and there are around 8 individuals who have spondyloarthritis in that family. So it's called a multiplex family, so multiple members of the same family. So if you study members of the same family, they have a lot of gene sharing. So if you look at what genes are abnormal or different from those individuals who have ankylosing spondylitis versus those who are normal, you will have very small number of genes that are different. And perhaps you can pick up the abnormal genes that are linked to the disease easier compared to studying like a population with totally different genes, right?
Oh,
so it's very difficult to identify. So you have greater gene sharing, so it's easier to identify what is different and what may be linked to the disease. So that's the principle behind studying families. So in this particular family, there are multiple individuals who have the combination of a deletion in a particular gene, and they also have HLA-B27. So this molecule is involved in transporting B27 from inside the cell to the surface for that antigen presentation that I was talking about. The vast majority of individuals who have this combination of this gene abnormality with B27 have developed spondyloarthritis in one form or the other. So it looks like this combination may lead to significant, increased risk of disease. So we are looking at what exactly does this particular gene do, why would the transport lead to these abnormalities, and we are looking at what all other proteins are interacting with this particular molecule. So if you identify what the interacting proteins are, we might identify a lot of other pathways that are involved in disease processes.
Will that lead to new treatment options, or is it more pointing towards being able to cure?
That's the problem, right? So the difficulty with immune-mediated disease and rheumatic diseases there's a lot of overlap between diseases and also with normal processes that are required for the body. So it's always a balance between how can we modify the disease versus how safe can we treat the patient, right? For example, if you take out one of these antigen processing genes, you take out the ability of the body to process the infection-related genes as well, the infection-related molecules as well. So you don't want to completely take it out, and maybe finding the right balance. There are currently efforts to modify the expression of these genes, and studies are being done in animal models. It has not reached human trials yet, but if it's successful and promising there, we might see studies coming over to the human side as well.
Oh, that's interesting. Is there another research project you are involved in that you'd like to mention?
Right. So recently we had a study looking at how to identify sacroiliitis, which is one of the most important findings in ankylosing spondylitis. So sacroiliitis in individuals who already have had CT scans for other reasons. So Dr. John Chan, who is a rheumatologist, is a consultant in University of British Columbia, Vancouver. So he spent a year with us and he did a fantastic job looking at how best to identify and differentiate sacroiliitis in individuals with ankylosing spondylitis versus the sacroiliac joint changes that might be seen over a period of time due to wear and tear. So, as you know, inflammatory bowel disease, these patients who have inflammatory bowel disease undergo a lot of CT scans to identify the bowel disease. So, if you look at these CT scans carefully, you can actually see the sacroiliac joints. You don't need to do an extra CT scan. And around 10% of inflammatory bowel disease patients have ankylosing spondylitis. So if you carefully look at these CT scans and use this scoring system developed by Dr. John Chan, perhaps we can identify these patients much earlier than the usual 7 to 8 year wait time seen in patients for days. Yeah, so that's a new scoring system that we have developed.
That's wonderful. And why is it important to identify patients early? I know this is part of your research as well. Why is it important to identify?
Good, when we talk about early identification, when it comes to, for example, a pneumonia, we're talking about hours, right? Hours or maybe maximum days early identification. When it comes to ankylosing spondylitis, we're saying if you identify them within the first 5 years. Yeah, so that's the difference in magnitude, and you talk about early disease identification. Because if you go back a few decades, the disease was considered to not exist in females, right? The ratio was considered to be around 10:1, male female, and then a few years back, it got corrected to say 3:1, and now with the MRIs and all, and with the advent of the non-radiographic spondyloarthritis, it seems to that more females have non-radiographic spondyloarthritis compared to males. So the delay in diagnosis in females could be up to 20 years. That was the situation before. And when you look at the previous studies of TNF inhibitors and their effect on bone formation, there was no difference. If you look at just 2 years of TNF inhibitor treated patients and 2 years of standard treatment like anti-inflammatories and exercise, there was no difference in the rate of progression. But now with earlier diagnosis and our large cohorts that we have a large group of patients who were diagnosed earlier than 5 years, 3 years, that range compared to those who were diagnosed after 10 or 15 years. And if you look at the rate of progression in these two groups, those who were diagnosed early and treated with TNF inhibitors had a much lower rate of progression compared to those who were identified late. So clearly there is a window of opportunity. The question is, what is, where is the point or what point defines the tipping, from no progression to progression? We still don't know. I would say the earlier the better.
The earlier the better.
Yeah.
I agree, because it's a terrible thing to live with a disease undiagnosed. I'm just wondering if there's any questions from our audience. I know it's terribly early in the morning for questions, but Anita, do we have any questions?
So someone wants to know, is the window of opportunity different for everyone?
Good question. So there are some predictors of progression that we can apply here. For example, as a person who is a smoker and already has some kind of damage in a baseline, and those who have elevated inflammatory markers like ESR and CRP, at least in one study it's 50 to 55% increased risk of progression. Compared to somebody who doesn't smoke and no inflammation, it's like 4 or less than 5% chance of progression. So the window of opportunity for a smoker and those who have a lot of inflammation may be different from those who don't smoke. Similarly, it is considered that females progress at a much lower rate. Perhaps the B27 gene is important. So if you take all of these factors together, I'm sure the window of opportunity will be different for different people.
Oh, thank you. And any other questions, Anita?
Is the treatment for female and male the same as of now, or is there something that works better in female patients versus in male patients?
That's an excellent question. So currently the standard of treatment is based on the presentation and the major symptom. So if you have peripheral arthritis, it is treated differently from spinal involvement. For example, methotrexate, sulfasalazine, these are disease-modifying agents. They are effective to an extent for the peripheral arthritis. We have not been able to prove that they are effective in the spinal disease, although a few patients do come back and report some improvement. It is not considered to be an effective treatment for spinal disease. That's really the only difference that when we approach treatment for spondyloarthritis patients. But the effect of treatment is slightly different. So men tend to respond better to TNF inhibitors compared to women. Having said that,
interesting.
Yeah. We don't completely understand why. If you look at the number of TNF failures, it appears to be, the results are better with males, with more failures in females. But it's not a huge difference. But we don't select treatment based on the sex or gender of patients.
I see. Well, that's wonderful. Thank you. I've learned something. I hope you have too. And I'd like to thank Nigil Haroon for two things, really. One, for coming for this interview, and two, for the incredible work he does at the cellular level in understanding spondyloarthritis. Thank you, Nigil.
Thank you very much.
Great for you to come in.
Thank you.